Assalamualaikum and hi there everyone. My name is Dr. Mardiana Abdulaziz from the Department of Pathology, University Technology Mara, UITM. This is part one of a series of lectures which will cover the topic of acute inflammation. So what will be covered in this part of the lecture is as follows. So at the end of part one of this lecture, you should be able to define acute inflammation, briefly describe the causes of acute inflammation, list the cardinal signs of acute inflammation, differentiate between exudate and transudate, and explain the pathogenesis of acute inflammation, which encompasses vascular and cellular events. Okay, so have a quick look at these images and see, have you ever experienced any of these? I'm sure you have. So if you look at the left side, you can see that the eye is very, very red. And if you recall the experience yourself, you might remember that when you wake up or when you woke up in the morning, you noticed that your eye was really sticky, kind of difficult to open because there was this yellowish exudate kind of sticking at the edges of your eye. So this is conjunctivitis or inflammation of the conjunctiva or the eye. And on the right side, okay, well, I'm sure you have experienced this before, where you got this redness and a little bit of bumps and swelling due to mosquito bites. Okay, so these are examples of manifestations of inflammation. So what is inflammation? So inflammation is literally a host response which aims to deliver leukocytes and plasma proteins from the circulation to the site of injury. So this can only occur in living tissues. So it will not occur after death. It is technically a protective mechanism which aims to get rid of, number one, the cause of cell injury. So this could be either microbes or toxins. And at the same time, it also aims to get rid of the consequences of that injury. Because sometimes as a result of inflammation, you can get necrotic cells and tissues, and these need to be eliminated as well. And in general, when there is an inflammation, we would use the suffix itis to denote inflammation of the organ or tissue. So as the example that we've seen earlier, inflammation of the conjunctiva, we call it conjunctivitis. If you have an inflammation of the appendix, then you call it appendicitis, and so on and so forth. Causes of inflammation are diverse. And examples include any sort of immune reactions or hypersensitivity reaction, such as autoimmune disease or allergies, or could be due to infection, which is probably one of the commonest causes of inflammation. It could also be due to tissue necrosis, which can be a result of trauma or some kind of physical chemical injury. And it may also be due to the presence of foreign bodies, which may be exogenous coming from outside, such as sutures or splinters, or endogenous, internal to the body, such as cholesterol or urate crystals. So depending on the cause of inflammation, the reaction may be in the form of an acute inflammatory process or a chronic inflammatory process. There are several features that differentiate between the two, such as the onset or the duration, the predominant cellular infiltrate, the tissue injury or fibrosis that occurs, the presence of cardinal signs, and the systemic effects of inflammation. So in today's lecture, we'll focus on acute inflammation, and then we will have another set of lecture for chronic inflammation. So you can see the features there. They tend to have very short, a quick onset, short duration, and lots of neutrophils, very mild and self-limited, typically prominent cardinal signs, and may have systemic effects. So mentioning about cardinal signs of inflammation, just to list them here. So cardinal signs of inflammation refer to the local manifestations of inflammation at the site of injury. So if you recall any sort of superficial inflammatory process that you have experienced yourself, you will have redness of the area. It will appear red. It will be warm to touch. Maybe there will be swelling because of edema. You may have felt a lot of pain, and at the same time, you may have lost function of that particular tissue or organ. We'll go through a little bit more detail in their pathophysiology in the second part of the lecture of acute inflammation. Okay, so now down into the pathogenesis of acute inflammation. What actually happens in an inflammatory process? Okay, so number one, of course, you will have that injury or infection or trauma, etc. So that is your offending agent. So the first thing that happens is your body will recognize the offending agent, and that leads to the process of recruitment of leukocytes and plasma proteins to the site of injury. And this occurs via a sequential step of events which include the vascular events as well as the cellular events, which result in the leukocytes and plasma proteins being at the site of injury, where they will be activated and they will perform actions to eliminate the offending agent. After that is sorted, then you will have termination of that inflammatory response, and that is followed by repair of the injured tissue. Okay, so I mentioned earlier about vascular and cellular events of inflammation. So here I'm just going to list down what they are. Vascular events refer specifically to changes of the vascular caliber. And when we talk about vessels involved in inflammatory process, they're not the large muscular arteries or vessels. They are the small vessels at the site of injury. So what happens in the vascular event of inflammation is that, first of all, these small vessels will dilate. So the diameter will be larger, and this will increase blood flow to the area. This is followed by an increase in the vascular permeability, which will allow leukocytes and plasma proteins to leave the circulation and again go to the site of injury to perform their function. And the third thing that occurs in the vascular event of inflammation is there will be an increase in lymphatic flow, and this will assist drainage of any extravascular fluid, so drainage of any edema fluid. And at the same time, because this fluid goes to the lymph nodes, it will also help in antigen presentation and further stimulate the inflammatory response. The cellular events refer to events which involve the cells of inflammation. So these typically are the leukocytes. Okay, so what happens is that there will be margination of the cells, okay, towards the endothelial surface of the vessels. There will be adhesion of these cells to the endothelial surface. They will transmigrate, so cross through the vascular wall to basically move from inside the channel into the extravascular space. Then, with the process of chemotaxis, they will migrate and move towards the site of injury, and at the site of injury, they will be activated and they will perform their functions in terms of phagocytosis, intracellular killing, and degradation. Okay, so to illustrate them a little bit better, let's have a look at these images that follow. If you see up here, this is what normal vessels will look like. Narrow caliber and only occasional resident lymphocyte or macrophage sitting in the extravascular space. With inflammation, the diameter of these vessels will increase, and this is vasodilation, which leads to increase in blood flow. The increase in blood flow will cause redness and warmth, which is part of the cardinal signs of inflammation we mentioned earlier. On top of that, there will be increase in permeability of these vessels, which allow plasma protein and leukocytes to escape and exudate into the extravascular space. Okay, so this is also responsible for the cardinal sign of swelling or edema. Once the fluid and some of the cells have escaped out into extravascular space, this will lead to loss of fluid within the vessels and lead to concentration of red cells. And this is what we call stasis of the blood flow. And this also contributes to the redness that you see at the site of injury as part of the cardinal sign of inflammation. So this is a vascular event in inflammation. What is not shown here is the increase in lymphatic flow. Vasodilation is mediated by mediators such as histamine, prostaglandins, and nitric oxide, while increased permeability is a result of actions of some mediators such as histamine, serotonin, C3A, C5A, bradykinins, etc. Increase in vascular permeability is one of the key vascular events in inflammation. So how does this happen? One of the principal mechanisms is by retraction of the endothelial cells, which allow gaps to form in between the endothelial cells, and this leads to a space where leukocytes and plasma proteins and fluid can escape out into the extravascular space. Another mechanism is endothelial injury, in which where the endothelial cells themselves are damaged, these cells can shrink and they can also form gaps in which the leukocytes and plasma proteins can escape out. So this process is called fluid exudation. And the fluid that exudates out and collects in the extravascular space forms what we call edema. Edema in inflammation tends to be of exudate type, where they have high protein concentration, they have lots of cellular debris, and they have high specific gravity. And this is because of the increase in vascular permeability allows more than just plain fluid to escape out, but also all these other constituents. And they contrast with the characteristics of transudate. When edema forms and collects in the extravascular space, the lymphatic system responds by increasing the lymph flow to drain this excess fluid into the lymphatic system. Let's move on to the cellular events of inflammation. Following the vascular events of inflammation, fluid loss into the extravascular space will lead to stasis and slowing down of the blood within the vascular channel. So more white cells will accumulate along the endothelial surface in a process called margination. So you can see in the image here, the leukocytes are lining up on the endothelial surface at the edges of the blood vessel. So this is the first step in the cellular event of inflammation. So once they have marginated to the surface of the endothelium, they will begin to roll. So they roll on the endothelial surface, and subsequently they will form brief adhesions, transient adhesions, and eventually they form stable adhesion onto the endothelium. And this happens first of all by activation by chemokines, so the integrins as well as the selectins are activated. So this is where the leukocytes bind to, and eventually the stable adhesion is formed by binding of the leukocytes to these integrin ligands. And eventually, once they have formed the stable adhesion, they will migrate through the endothelium, as you can see here. Transmigration is also known as diapedesis, and it is the process where the leukocyte will migrate through the endothelium into the extravascular space. In the extravascular space, they will move towards the site of injury in a process called chemotaxis, and this is by definition locomotion along a chemical gradient, also known as chemoattractants. The chemoattractants can be exogenous, so something coming from outside, such as bacterial products, or they can be endogenous, such as cytokines or components of the complement system. The process of chemotaxis will bring the leukocytes towards the site of injury, where they will recognize the offending agents and they will be activated to perform their subsequent function. So this is what chemotaxis looks like. So these red dots or lines represent the chemoattractants, and you can see that this is a leukocyte which tend to be attracted and move towards the site of the highest gradient of the chemoattractants. So when they are activated, then they will perform the next step, which is phagocytosis and then intracellular killing as well as degradation of the offending agent. So phagocytosis and all this intracellular killing involves several sequential steps such as follows. So imagine you have the offending agent depicted here is infection. So there is a microbial product which will be recognized by the leukocyte. So the bacterial product will adhere to the phagocytic receptor. The phagocyte or the leukocyte will engulf, okay, so they form a membrane all around this microbe which is now attached to the receptor, so engulfment, and that forms like a vacuole inside the cytoplasm of the cell, and this is called phagosome. Okay, so this is called phagosome, and now the microbe or whatever agent that's causing the injury is now phagocytosed and located within this phagosome. In the cytoplasm of leukocytes, there are also lysosomes. Okay, so lysosomes are separate vacuoles which contain enzymes which also have the capacity to degrade and kill microorganisms. So now the phagosome, which contains this organism, as we show as an example here, will fuse with the lysosome and it forms a phagolysosome. So all the activities of intracellular killing or degradation will occur within this membrane, which is sequestered and separated from the cytoplasm of the cell. Okay, so this ensures that the cell itself will not be damaged in the process of phagocytosis and killing. Okay, so recognition and attachment, engulfment, and then you have fusion of the phagosome with lysosome, and now you get the killing and degradation within the phagolysosome. And the killing and degradation takes two forms. The main form of killing is usually by the production of reactive oxygen and nitrogen species, and a secondary method of killing is by direct action of lysosomal enzymes. So in the phagolysosome, you can have production of reactive oxygen species by formation of hydrogen peroxide, and these can react with myeloperoxidase in the presence of halide, and they are converted to hypochlorite, which is highly toxic to the organisms. Reactive nitrogen species are produced as nitric oxide reacts with superoxide to form the nitrogen-derived free radicals. So again, they have the capacity to kill microbes and degrade the offending agents. So another method is by the action of lysosomal enzymes or proteins, either primary granules such as myeloperoxidase or secondary granules such as lysozymes. So they also have the capacity to kill and degrade. So once all that is sorted, then acute inflammatory response will be terminated. And there are several factors that help in the regulation and termination of an acute inflammatory response. So number one, it is the characteristics of the mediators that are involved in the process itself. Okay, so the characteristics of the mediators can help in this process because they're produced in rapid bursts, so they can be terminated very quickly. They are only present for the duration of the persistence of stimulus. So once the stimulus is gone, then the mediators will no longer be produced or no longer be activated. The mediators tend to have short half-lives, so they can be degraded quickly as well. And in the process of acute inflammation also, the main cells are neutrophils, And neutrophils have very short half-lives in tissues. They only live for hours to 1 to 2 days. So they will be gone quickly, and thus the inflammation can also be terminated. On top of that, there will be active termination mechanisms. So this can be in the form of switching in the type of metabolite produced, and also in the form of actions of anti-inflammatory cytokines. So we talked about how acute inflammation is actually beneficial, is something that we want to happen, but at the same time, you can get side effects of it, the not so desirable effects of it. And this includes the possibility of damage of normal tissue because of digestion of normal tissues by release of lysosomal enzymes from the inflammatory cell. You can have complications from all the events that occur in inflammation, such as edema or swelling. So whenever somebody has a swelling or edema, depending on the location, it can cause some problems, such as obstruction of the airway, increase in intracranial pressure, etc. And sometimes the inflammatory response is inappropriate. You don't want it to happen, but it happens anyway, such as in an allergic inflammatory response in asthma. So that concludes part 1 of the lecture on acute inflammation. So to summarize, in this part of the lecture, we have covered the causes of inflammation and also the pathogenesis of the sequence of events that occurred in inflammation, which include the recognition of the offending agents, the various vascular and cellular events to recruit leukocytes and plasma proteins to the site of injury, its subsequent activation, phagocytosis, intracellular killing and degradation, followed by termination of inflammation and repair, which we will cover in our subsequent lecture. Thank you.